But just to begin with a very short sort of overview of the company, so Horizon Discovery, we're a biotechnology company. We're based in Cambridge, UK, and we were founded in 2007 as a sort of translational genomics company. With a core expertise around genome engineering and cell line generation, and in particular, we've been offering products and services in the research sector surrounding these isogenic cell line products. So initially we use a lot of RNAi to generate these cell lines, but more recently we've invested very heavily and very interested in nucleases like Cas9 and zinc finger. But we offer products and services from the beginning to the end of research services. So the overall the model would be that the difficulties and the escalating costs of drug discovery programs and the failure of the blockbuster model for drug discovery has resulted in a really exquisite need for new tools that can reduce costs and provide better insights early on in drug discovery. And we think this is what our isogenic cell line models can do and the products that arise from them. So let me move on and then give a little bit of an overview of the sorts of services we provide. We provide products and services in vitro and in vivo, all the way from very basic, simple research questions through even to more recently drug manufacture in our bioproduction program and even molecular diagnostics with our reference standard provisions. But today I want to talk about functional genomic screening, which is a service that we launched very recently and that we're quite excited about. So let me just give a little introduction to the agenda for today. So I'll give an overview of the CRISPR-Cas9 sort of biology and how this applies and how we're using it in functional genomic screening. And then I'll try and introduce some of the improvements that Horizon have made, what we've learned from our experiments so far, and seeing where we can really apply this technology in the future. And then I'll finish with just some forward-looking ideas and some of the more recent things we've been doing and looking to do using this technology. So to begin with functional genomics itself, so this is the coupling of genetic information to phenotype, but where functional genomic screening comes in is really to start to look at a more high throughput, potentially even unbiased analysis of linking genetic information to really cellular phenotype. And there's some examples of where this might be applicable highlighted on the screen in target identification, in drug mechanism of action, patient stratification. These are very much pertinent and important areas of research, but really functional genomic screening is really very widely applicable throughout biology, really. So it touches on a lot of different aspects for people's research. I'll use the examples highlighted on the screen today as sort of touchstones and as exemplars for how we've been analyzing the technology and how we think it can be used largely in oncology, but I do want to stress that we think that there's a lot of opportunities outside this sphere of interest. So functional genomic screening at Horizon has been going on for several years. We started our interest looking primarily at RNAi and in specifically siRNA arrayed screening. Our interests were particularly in identifying new synthetic lethal targets. But we recently added a gene trap screening program to this, which is used explicitly in haploid cell lines, and that's a complete knockout program, which is, we had a webinar about that just a few weeks ago, so that should be available on the website. But today I want to talk about CRISPR-Cas9. This is a pooled screening approach, not dissimilar in its infrastructure to shRNA screening, but very different overall in terms of the quality of the data that you're able to generate. So this is the third arm of our functional genomic screening portfolio, and the one we're really excited about and are going to talk about today. So then just a little bit about RNAi and why we decided we needed to find complementary methods to look in more detail about the capabilities within functional genomic screening. So RNAi has got some really important advantages, but it's got some well-known limitations as well, and I've highlighted a few of these on this slide. So firstly, we know that RNAi is limited by its partial knockdown in terms of its actual overall phenotypic range. And this is highlighted on the left-hand side, where we're looking at, actually this is from Feng Zhang's paper early last year, looking at cytometric outputs from knockdown of GFP, and you can see that there's 4 hairpins shown, and really none of them show a significantly greater knockdown than around 70%. Now, that's useful sometimes. Some phenotypes will only be elicited with modest amounts of remaining expression, and also maybe this simulates druggability quite well, but it doesn't give you the maximum phenotypic window. So it isn't actually very useful when it comes to large screening efforts where quality and signal to noise is really important. And then secondly, in the middle, RNAi has been plagued by low reproducibility between screens, even run in parallel in the same labs across continents. It's been very difficult to identify systems which will allow high reproducibility with RNAi. That's highlighted in the middle. There was, in this example, there's 3 independent studies looking for HIV host factors. Each of them identified around 200 to 300 potential targets. Only 3 of them overlapped. I'm not sure how many of them even validated at the end of it. So it's really difficult to show overlay between independent experiments with RNAi. And then finally, on the right-hand side, off-target effects are very well known in RNAi. This is an example that comes from a paper published this year when we were looking at, a group were looking at correlating the phenotypic response with transcriptomics to the sequence of an shRNA, or rather the target, and comparing that to the seed sequence. So that's the short truncated fragment of an RNAi antisense fragment, which results from endogenous processing of the sh or siRNA. And that's a frequent cause of off-target effects. And remarkably, what this group actually found was that the phenotype that they're observing was more frequently correlated to the seed sequence than it was to the actual target, telling you that most of the effects were caused by off-target effects. So that's a really difficult thing to overcome. It's inherent to the technology, and it's something that we're looking to improve upon with the CRISPR-Cas9 approach. So what is CRISPR-Cas9? CRISPR and the Cas9 endonuclease were discovered several years ago. They provide an innate immune system in bacteria, but they've been now really repurposed and engineered to a certain extent to be used in all sorts of new and exciting experiments. The basic principle is that you have an endonuclease, which is Cas9, and it forms a ribonuclear complex with a short RNA fragment. In our case, this is a chimera of a scaffolding domain and a targeting sequence, and we call the whole thing a guide sequence or an sgRNA. And together, these 2 components come together to target the endonuclease to a specific genomic loci. And at that loci, then the endonuclease, which has 2 independent catalytic active sites, can then introduce double-strand breaks at the precisely defined locus. Now, in mammalian systems, what that results in is the repair mechanism, NHEJ, non-homologous end joining, of that DNA strand, which is inherently inaccurate. And consequently, you get frequent introduction of insertions and deletions, and many of these result in knockout of the gene due to out-of-frame mutation. So what this allows us to do is to essentially, if you target, that means the endonuclease, early enough on in the open reading frame of the gene, then you can knock out that gene completely. And this works across multiple alleles and provides a really robust phenotype due to that complete loss of gene expression. So we think this has got a real strong advantage, not just in genome engineering, where we also use it, but also in looking at functional genomic screening. And in particular, our interest in providing fewer off-target concerns than RNAi. Now, this is an emerging field of study, but it looks strongly likely that this is going to be the case. And actually, the way we design our screens, as I'll describe on the preceding slides, to some extent mitigates any off-target effects anyway. Just to give then a timeline, actually, for how long we've been interested in CRISPR and where we started with a CRISPR screening approach. So 2013 was the publication of some of the most important papers in CRISPR, the translation of that technology into mammalian cells. We started working on CRISPR for engineering quite quickly, and then the screening papers were first published towards the beginning of 2014. That was from Feng Zhang's laboratory and from the Landers-Sabatini groups initially. And we were immediately interested in this and have started generating the necessary infrastructure pretty quickly after that. It took us about a year to do so and to get our first screening data sets, and since then we've been optimizing the process, gathering new data, and even deploying it now in some internal screens. And that has led to the present situation where we've launched this as a screening service just in the last month or so. And I'll give details about that service towards the end of this talk. But now let's move on to talk about the actual screening process. So the screening process, as I mentioned, is not dissimilar to pooled shRNA screening, but it really begins with the definition of what the experimental outcomes that you desire really are. So that means selection of a number of genes that you are interested in targeting. That might be completely unbiased, a whole genome screen, or it might be a specific subset of genes that you're interested in targeting. And then from that, you need to then design a library. I'll talk a little bit about library design later on, but usually it requires the introduction of minimally 5 to 10 independent sequences targeting every different gene that you're interested in the library. Then you can pick your cell line, and it may require optimization. It's very important to optimize the cell line and make sure that the conditions that you need for the transduction program are optimized for that cell line. Because once the library is designed, you then need to generate generate the sequences through massively parallel array-based synthesis, a bulk cloning reaction, and then make a lentiviral particle pool. The cells are then transduced, and the transduced cell lines will be selected either through antibiotic selection or through fluorescence, and then screen assay conditions can be introduced. So that might be application of a particular drug or a growth condition, or just time to allow a phenotype to be manifested. Then all of the analysis is done with deep sequencing, and what I mean by that is that we use a fairly sophisticated way of mapping the sample populations in a quite high depth. So minimally, we'll have a lentiviral library or a plasmid library, and we'll have control samples and a number of different test samples. But in essence, each of these are treated in the same way. And in each case, you need to collect the genomic DNA and deep sequence it. And what you're sequencing is the lentiviral cassette itself. So that's introduced into the cell line stably, and we sequence the sgRNA sequence directly, and we use this as a barcode. And that barcode tells us what that cell line was knocked down for. So it's a proxy for the genotype, effectively. So once we map the guide sequence using deep sequencing to the desired gene that it was knocking out, we can then quantitatively determine what the population was in each of those different samples for those different guide sequences. And in that way, we can then interpret the phenotype of that cell line using this as a mapping strategy. So what sort of screens can we do? So the first examples of screens are the simplest, really. These are positive and negative selection screening. And this means that once you get your cell line, which is, of course, carefully optimized for conditions for screening, you can then introduce the lentiviral library to this cell line population, and then introduce, as I said, screen assay conditions. In the case of positive selection, what you're looking for is genes which, when knocked out, confer a resistance to that condition. So these will be enriched in the population. They'll overcome the cytotoxic consequences of the drug treatment, and then at the end of the screen, you'll be able to collect them quantitatively as having competitively outgrown other cell lines in that culture. And so this is what you'd use for resistance screening. You'd use that potentially to gain insights into mechanism of action of a drug, or to understand a priori potential resistance mechanisms of a compound prior to it going into clinical studies. On the other side, you've got negative selection, which is where you'll be looking for genes which, when knocked out, they'll actually confer a selective growth disadvantage to those cell lines, so those individual cells. And that will allow you to find genes which sensitize your cells to a certain condition. This is much more difficult to do because you're looking for things that are lost in the population rather than enriched, so they're inherently more difficult to identify. But the principle is not that dissimilar. You may set up these screens in different manners, because in a positive selection screen you might want a high concentration of compound, whereas in a negative selection you'll want a subcytotoxic dose. And in that way you'll have to decide differently how to design your experiments. An alternative approach in the screening application is to look at genetic interaction studies. So in the simplest case here, you take, instead of a single cell line, you take several cell lines. For example, a cell line which is wild type at a particular locus and a cell line which is a mutant, and you'd introduce a library targeting a collection of genes into both of those cell lines. You then grow those cell lines out so that you can manifest the phenotype, and then look for differential responses in the two populations. So, for example, where a gene knockout was particularly lethal in the mutant cell line, that would tell you potentially that you've identified a synthetic lethal partner, which is selectively detrimental to the growth of a particular genotype. Actually, at Horizon, we take this a bit further and we look not just at 2 cell line collections, really we look at multiple cell line collections, and in each collection we're looking at multiple cell lines that represent that genotype. And that really increases the potential power that you have to draw out potential targets from that program. So now let me give you a few examples of some of the data that we've been generating, both as proof of principle and some novel data sets as well. So the first is a screen for resistance mechanisms, and this was done in A375 cells. These are a BRAF V600E mutant cell line, and what we were looking for in this experiment is to identify factors which, when knocked out, confer resistance to the BRAF inhibitor vemurafenib. And the data from this are shown on the left-hand side. So this was a whole genome screen, so that means it targeted around 20,000 genes. And what we're plotting on the y-axis is the significance factor, and on the x-axis is essentially the enrichment factor. This is in log2 scores. And what's highlighted are a number of genes we identified as hits. What's useful about this screen is that it's a recapitulation of a couple of other screens that have been done. A similar screen was done with RNAi, which identified 2 potential hits for vemurafenib resistance. And last year, Feng Zhang also published a screen using CRISPR-Cas9 in exactly this paradigm, and he was able to identify a further 4 factors which conferred resistance to vemurafenib. Our screen was able to identify all 6 of those previously known and validated targets, and an additional 2 factors which from the same gene families. So these are genes typically which regulate MYC or MEK pathway, and are well-known pathways for enacting resistance to vemurafenib. So this is a really good proof of principle for the CRISPR-Cas9 screening at Horizon, but more importantly, actually, it really validates the technology overall, because this is a direct recapitulation of a screen previously done, albeit with a library that was at least 50% different to that which was previously used, and yet it came up with exactly the same hits with really, really high power in the identification of those hits. That's a really nice proof of principle study. On the right-hand side is a bit more depth to that data, where we're looking now at the individual guide sequences that target each of those genes from that hit. So I mentioned that you would use multiple guide sequences to target every gene. In this library, there are 6 guides which target every gene, and the enrichment scores for each of those guides from those genes are plotted on this axis here, which shows the enrichment factor. So this is a log2 score, so you can see that actually in some cases these genes represent cells which were enriched by up to 2,000-fold in the population overall. But it also shows you that some of the guides don't work at all. So those are guides which do nothing, even though you'd expect the majority of the guides that were functional to elicit a phenotype. And what that tells you is that some of these guides are indeed inactive, and that's what you expect in this study, because the library design, the information required to make perfect guides every time is not yet available. But actually it doesn't matter, because you target the gene with enough guides so that you can still identify the genes that are hits. And the most interesting thing is that these guides do indeed cluster around the 0 point. There's 1 or 2 that show some depletion and maybe a little bit of off-target effect. But overall, the noise in the system is extremely low, and the quality of the data is very, very high. So that's a really exciting observation, and it opens up lots of different possibilities. One of those possibilities is then to look in the more difficult, challenging paradigm of sensitivity screening. So here, instead of looking at things which are enriched, we're looking at things that are lost from the population. So that is cells which, when knocked out for a particular gene, are selectively diminished in the population overall, if you take from time 0 to time n in a given screening condition. So in this example, we're looking again at whole genome screening in the same library, this time in haploid cells, and we're looking at glucose starvation as an experimental paradigm. So this is taking genes which, when knocked out, confer selective sensitization to loss of glucose in the screening media. And what we identified is a number of NADH dehydrogenases and complex 1 components, which is exactly what you'd expect to see from a screen of this sort. We also identified a couple of tumor suppressors, which are cytoprotective under conditions of stress. So again, loss of these genes would be expected to sensitize cells to a stressful condition like glucose starvation. So that again is a really good proof of principle that sensitivity screening, albeit challenging, is possible to do using this approach. So I'm going to move on now to talk about some of the more exciting discoveries and some of the questions that we had that we needed to address to really assess the power of this screening platform. So we were looking to design a series of experiments that would help us to improve the system and understand more about CRISPR-Cas9 screening in general. So we wanted to know, in particular for dropout screening, so sensitivity screening, to what the magnitude of a dropout you can expect to find from a real essential gene would be. So we know how to classify what would be a real hit and what isn't a real hit, and know whether or not also we can improve this. We're also really interested in library design itself and the complexity required of a library to gain significant hits from the screening exercise, and also understand more broadly about how reproducible these screening programs are. We're also interested in the time resolved aspect of this CRISPR-Cas9 function. So really, what the kinetics of the enzymatic function of Cas9 are, and how this is elicited throughout a screening program. And finally, we're interested in the sequence-specific determinants for guide design. And this is a program we were conducting in collaboration with our partners at Desktop Genetics. I'm not going to talk about that data today, but hopefully there'll be something coming soon in the pipeline. So the basic program is that we would use essential genes, so that is genes which we know to be essential, as a way to monitor CRISPR-Cas9 activity. So that means when you knock out a gene which is known to be essential, then we should be able to see it lost from the population over the duration of the screen. And in that way, we can map back the identity of that guide or that gene and use it as a way to monitor the efficacy of the CRISPR-Cas9 screen. So the first bit of data I want to share is an overview of what the results from that experiment were. So this is showing a number of different collections. 3 independent collections of essential genes are shown in the pairs of bars on the left-hand side, and then some neutral or non-targeting genes are shown on the right-hand side. And the first interesting thing to note is on this final set of bars that I'm highlighting now with the laser pointer shows you non-targeting guides. So these are essentially guides which shouldn't target anything anywhere in the genome, and these are modestly enriched in the population. And we think that's a biological phenomenon because these guides don't cut anywhere in the genome, so consequently no DNA damage response is elicited, and they confer a modest competitive advantage in the collection of cells that you're analyzing. So they slightly enrich over time. The second thing to note is that all of the collections of essential genes are indeed depleted in the population. So what we're plotting on the left, on the y-axis, is the relative abundance of these guides and the change of that over the duration of the screen from time 0 to the end of the screen. So a drop down from 0 then indicates that they were lost relatively in the population. So it indicates that they were indeed essential, which is exactly what we were hoping to see. But the most exciting thing is that we were using this system to monitor the performance of a new optimized screening backbone system, which is shown in orange and compared to the older system, the p-lenti CRISPR V2, which is shown in blue. So in this collection of bars here in the center of the screen it shows actually the ribosomal genes, and what you can see is in this case the overall dropout for these genes when you use the new vector system is in the order of 10-fold. This is a log2 scale. So that's a hugely significant improvement in the system, and it opens up, as I mentioned earlier, really exciting new possibilities for using this technology in dropout screening. But just to look in a bit more detail at that result, this is now showing all of the individual guides overlaid just for the essential genes, and now overlaid one on top of the other. So everywhere where the new vector system outperforms the old vector system, you see the orange bars dropping down, and now you can see that a really, really significant proportion of them are of massively improved performance. And now you're looking at the individual guide level, you can start to see that the dropout rates for some of these guides, are in the order of 1,000 to 2,000-fold. So really, really significant. Whereas in the old vector system, you're really only getting a handful that drop out to that level of magnitude. So it massively improves the power of the screening, and that's of particular relevance to dropout screening like this. So now I want to move on and talk a little bit about library design. In this experiment, what we were looking to analyze is the difference between available library design systems. There are lots of library design systems available now. We were just looking at 2 of them. One of them is we call the Gecko V2 library guides, which were designed by Feng Zhang's lab in mid-2014. And then there was a second generation of guides designed by Landa Sabatini's group, which were machine learned from experimental data sets, essentially. And so they provided a potentially improve performance. There is also third and fourth generation library designs, and these will be improved all the time. But just to look at these two as an example, the first thing to highlight is that the Gecko library contains 6 guides targeting every gene, whereas the Wang library, which is the guides from Landa and Sabatini's groups, targets 10 guides every single gene in the collection. So there are more guides targeting here. And now I've just overlaid them. They're ranked from 1 to 0 independently, so they're not overlaid. But what you can see is that the guides which are ineffective, essentially, even in this essential collection, you really don't increase the number of those despite the increase in the number of guides targeted within that library. And what that tells you is that the machine learned set from Wang et al.'s paper actually provides fewer guides which are inactive, which is a useful thing to have. And secondly, you can see that there are substantially more guides which target with and result in significantly depleted levels of dropout in these in this experiment. So what I mean is that there are a greater number of highly active guides in the machine learned second generation library, which again is really valuable. So on the basis of this, it would appear that the second library, the machine learned data set, does indeed provide an improvement in the performance. But really interestingly, what happens when you look at these guides in our new vector design system, actually you can now see that both libraries perform relatively equally well. And that means that even guides which were only modestly effective from the Gecko library are actually significantly improved in their performance when you move them into the new vector backbone system. So that's really interesting and important because it opens up a greater number of guides that you can actually use to target. And it tells you that although guide design is really important, and of course there's lots more to be learned about making a really active guide and knowing about the sequence determinants and the non-sequence determinants of that design, actually the greatest impact you can have on the quality of your screen is to do the system with really carefully designed system and using the right infrastructure to do that. So that's also really important for us to know. Okay, so now I want to move on and talk a little bit about reproducibility. And we conducted screening in several different cell lines. And in this case, what I'm plotting is 2 cell lines overlaid over the top of one another. And again, this is now looking at essential and non-essential genes, so the whole population. And I'm plotting, in one case, a haploid cell line response, and in another case, A375, which is a triploid cell line response, using log fold change again as a measure of the efficacy of the guide. And the point I want to make with this slide is that the data that resulted from these 2 cell lines are really remarkably well overlaid, in particular with the non-essential guides, which would have varying phenotypes. The correlation is really close. I wanted to show this graphically, which is more interesting. But it really opens up really great new opportunities, in particular in things like synthetic lethality, where you're interested specifically in the difference between 2 cell lines. You need to have really high quality data, really low signal to noise, to be able to identify genotype selective differences between those 2 cell lines. And what this tells us is that if there was synthetic lethality within this gene collection between these 2 cell lines, which you wouldn't necessarily expect, actually it would stick out like a sore thumb. So that's really exciting. Okay, so finally then, from the data side, I want to just talk a little bit about the kinetics of CRISPR-Cas9 and how fast can a guide be expected to function. So this is an open question, and this is just one piece of data that we think is interesting to examine this feature. What you're looking at here is the counts overall for a collection of guides, all targeting ribosomal genes. So these are all expected to be essential, and you're plotting them through time. So essentially, the counts shown on the left-hand side are from the plasmid input. This is the time 0. And then from T1 to T4, you're looking at different time points within the screen. So if this is targeting a gene which is essential, you'd expect that to be dropped out through the population, as indeed most of them are. But now you can actually start to look at the kinetics of it. And the point to make from this is that actually there's not a great deal of difference in time between T1 and T2. And T2 is really actually, in some instances, for some cell lines, you might classify that as the end of selection. So that's really the earliest time point you might feasibly be able to take. But actually, in this instance, what you're able to see is that many of the guides have already started to enact a phenotype by that point. They've already dropped out by as much as 2-fold in some cases. So if you were to start your screen there, actually you'd already miss most of the phenotype that you wanted to count. So it's really important to understand how to design your experiment and how to analyze it to be able to make appropriate hit calling decisions when it comes to the analysis. Okay, so I think that's where I'll finish on the data side. And I want to now just talk with a few slides about where we're going next and what we're hoping to achieve. So far, we've talked primarily about doing screening based on viability as a measure for phenotype. And that's really exciting and interesting, but actually there are some more potentially complex screens that can be done that will allow us to analyze more interesting phenotypes, I should say. So Horizon are really well positioned to do this because we have a lot of expertise in genome engineering, so we can generate reporter cell lines. And we can then use these reporter cell lines in our screening programs. So for example, you might generate a reporter cell line which shows a fluorescent reporter for a given transcriptional response or protein stability. We can then use this as the basis for a screen, transduce that with our library, introduce it to assay conditions, and then use flow cytometry or even potentially another readout to sort those populations into responders and non-responders and use deep sequencing again to identify hits from that screen. That's something that we're working on now, and I think that's going to be a really interesting new arm to this screening. Secondly, I wanted to just talk a little bit about target validation. This is really important as a component to any screening program to know how you're going to identify the real hits from your screen. As I've shown, the data quality is very high emanating from these screens, but you still need to confirm that in independent experiments, any hits that come out of it. And here's an example of how we've done that in one of our experiments. So this is a screen looking at 6-guanine resistance in haploid cells, and this is a DNA damaging agent, and what you'd expect from this compound is that if you knock out mismatch repair genes, you would confer resistance to this drug, and that's a relatively well-known paradigm. Indeed, that's what we found. A bunch of mismatch repair genes came out as well-scoring hits, including MLH1 and MSH6. And we then used our off-the-shelf haploid engineered clones, which are knocked out for these individual components, to validate these hits. And what we're doing here is an array-based experiment of a dose response. And what we found is that indeed, as we'd expected to see, when we knock out these genes, we confer resistance to this drug treatment, and it's elicited with a shift in the IC50 away from the parental cell line. So that really validates these hits quite nicely and in line with the experimental program. So the next thing we're really interested in is actually repurposed CRISPR screens. We're really particularly excited about using CRISPR-A. So this is a repurposed technology where you use an adapted version of Cas9, which is fused, it's nuclease dead, but it's fused to a bunch, through various different approaches, to a bunch of transcriptional regulators. In the case of CRISPR-A, these are activating components. You then target those using Cas9 to a genomic locus, a promoter in particular, and use this to screen for gain-of-function mutations, because what this system will do is will upregulate protein expression. So that's a completely different paradigm for biological analysis and opens up new opportunities for different sorts of experiments. Okay, so finally then, just to talk a little bit about what we can provide as a service. As I mentioned, we launched this CRISPR screening service recently, and we can provide a complete workflow from start to finish. That includes library design, which is a really important component, and that will allow what we're really interested in providing is careful library design, including the introduction of the appropriate controls into that library, which will allow you to validate your guides and your screen data sets and understand how high the quality of the data is. That will run all the way through the cell line optimization and screen assay design, and we will also do all of the bioinformatics and provide hit identification services as well. So what we'd really deliver from that is, of course, all the raw and analyzed data, but importantly, we'd also provide our hit nomination. So that will give you a list of genes which you can start to think about for secondary analysis and either directly into target validation or secondary screening. We work with a bunch of different libraries that are available. Some of them are highlighted over here. We also have whole genome libraries and some that aren't shown here, and new ones are being generated all of the time. And of course, we can do combinations of these libraries, and we're also able to provide custom libraries for bespoke gene sets for different screening programs. So then I'm going to finish just to highlight again some of the reasons we think this is a useful service. I didn't get a chance to mention in detail, but we have a very strong intellectual property position. I think we're the only company possibly with all 3 licensed estates around CRISPR-Cas9. So that's really important, I think. We have a burgeoning data set now that allows us to design really good libraries and really good experiments. We of course have you could access our proprietary backbone, which provides really improved performance, and this complete workflow, which I think is really interesting too.